Hey everybody. So today or tonight or whenever you're watching this, I suppose, we're going to be going over chapter 10. So if you didn't get my message earlier, go ahead and check the syllabus. I updated the schedule to reflect a more realistic view of the chapters that we're going to be covering, and maybe even some select topics from some of those chapters. Alright, so chapter 10, biosynthesis of nucleic acids, replication. So we're going to be talking about, in this chapter specifically, DNA getting replicated. In the next chapter, we're going to look at transcription, which is copying DNA into RNA so that we can, down the road in the next chapter after that, make proteins. So this is going to focus on DNA, and we're going to look at primarily prokaryotes. We will make some comparisons between prokaryotes and eukaryotes. Your book will go into more details than I'll go into as far as those comparisons go. But prokaryotes are a little easier to study. They're obviously much smaller. They have a lot less moving parts. They're less complex. Without, just the more obvious differences like, not having enclosed membranes on the insides of their cells, or not having their DNA associated with histones. So it's a little easier to get all the big ideas and the big picture looking at prokaryotes. So we'll emphasize that and make some comparisons. All right. Okay, so yeah, we're pretty much going to cover all of these things. So DNA contains the information to make all of your proteins. It also has some other non-coding parts that do something that we're still trying to figure out what all that is. Every time your cells double, every time they, what's the word for it? Every time they replicate themselves, I think the term in biology is mitosis, when they undergo mitosis, they need to copy everything that's on the inside of their cell and double it so that they can split into two cells. And depending on what type of cell this is in your body, this can happen every 24 hours. It can happen up to 120 days. The process of splitting into two new daughter cells. Replication refers to then the doubling of the DNA. And there are some RNA players, some intermediates. And we'll see specifically that we're going to need some primers. Transcription and translation are the subject of the next two chapters. Transcription is making RNA from the DNA to be the intermediate for the protein, and then translation is taking that intermediate, that messenger RNA, and then making a protein out of it. So that's what the terms replication, transcription, and translation all refer to. For the most part, DNA is what is thought to be the starting point, and that information goes from DNA to then RNA to then protein. And for a long time, that was considered the central dogma, the main sort of belief. But they did find out by some retroviruses, I think AIDS is the HIV virus that causes AIDS. This was a retrovirus which didn't have its genetic information stored as DNA. It actually has RNA, and it takes that RNA, makes DNA out of it, and then uses that DNA to then transcribe and translate into proteins. So in that case, there's an enzyme that'll actually read the RNA and make DNA. And so whether it starts with RNA or it starts with DNA, we can say the genetic information is stored in these nucleic acids. And this is sort of that picture here. DNA can double, or it can be turned into RNA, turned into protein. RNA can also self-replicate. We saw that in, I think, the very first chapter we looked at. And then in retroviruses, we can even make some DNA out of the RNA. Naturally occurring DNA. is single-stranded. So ours has to protect itself, and so what it has is a complementary strand that binds to all of our nucleotides and gives us that characteristic double helix that we have, that we looked at, I think, in the last chapter. The difference between our DNA and a lot of the prokaryotes that have been studied to give us all the information we know is a little different. The prokaryote DNA is circular, and then ours is linear, and then again associated with proteins. And so not every type of replication is identical, and that's exactly what the book is trying to make here, the point that the book's trying to make. So we're just going to talk about some of the commonalities between all the different types and get the general big picture. And again, most of this that we'll talk about was discovered by studying E. coli. Oops. All right, so there are some challenges that are presented initially when you're trying to figure out how to copy the DNA. And first is, how do you unwind the strands? And once they're unwound, how do you protect those now newly exposed nucleotides from degrading enzymes, things that are going to come in there and attack them? There's also another problem related to the unwinding. If you've ever tried to unwind something that is, or pull apart two strands that are kind of connected together, it's going to lead to some supercoiling, where you start to pull apart the two strands. The twisting is going to become even more intense downstream, or upstream and downstream, and so something needs to take care of that unwinding and that winding strain. In addition to this, there's the problem with copying the DNA. The enzyme that we have that's going to go in there and actually put in the polymerase that's going to put in the new nucleotides and build the new daughter strand only reads one direction on the DNA, and it builds the DNA from what does it say here? From the 5' to the 3' end. And we'll look at the structure, I think, in a second and see why that's the case. But that means that while it's going down one side of the DNA, the opposite, the complementary strand, is actually headed in the wrong direction, and so it can't read that one at the same time and simultaneously make, a complementary strand for that one. And so because the direction of the two strands that it's going to use to basically double the DNA are anti-parallel with each other, that's another issue. And so we're going to look at how kind of nature has taken care of these issues. The last also being, what do you do about errors? Mistakes happen. Nucleotides get substituted in when they weren't supposed to, and so we're going to look at how some of these polymerases, these molecules that build these new DNA polymers, can actually correct some of the mistakes that get introduced into the sequence. Your book talks about semi-conservative replication. So what this means is essentially that when this DNA double helix splits into two new copies, each copy is going to contain part of the original. That's what that means. And so you can see it here visually. It's colored this red strand here. Both of these both of these DNA ended up with one of the red strands, and so then one of each of these strands is going to end up in each of the daughter DNA. They're not the daughter strands. I guess they are the daughter strands. Parent strand, daughter strands. So G1, you see here, ended up right here, and G0 ended up right here. So semi-conservative just means it ends up keeping half of what it started with originally. And they did find this out through a neat experiment using a heavier nitrogen. You can read about that in the book. It's very interesting. And it's talked about right here in this slide. They incorporated some of the heavier nitrogen into some of the DNA, and then using centrifugation, we're able to sort of track where it went. Yeah, anyways, you can read that. Okay. When replication starts, it starts at a place called the origin of replication, and it takes off in both directions. And we know this by looking at some of the experiments. It's not 100% of the case, but DNA replication does happen bidirectionally. So where the two strands were, and then I'm going to show a little before and after, then we get some replication origin here, so this little bulge, some unwinding has happened, and then replication is going to take off in this direction and in that direction. I probably could have drawn that in a different place. Sorry about that. And the thing that's going to be moving along here is going to be called the replication fork, because if you can imagine, after this gets replicated, we're going to have, actually I don't know if I can even draw this. Right, there's going to be a new strand, this one being the new strand, new strands. And so this little area right here is the replication fork, and it'll be in both directions. There'll be another one over here. Okay, so two replication forks, and these are where the new chains are going to be kind of growing from. It's called a theta structure because it kind of looks like the Greek letter theta. In prokaryotes, there's one origin of replication, one bubble. They have circular DNA, so what happens is you get that little replication fork, and then at some point you got this going on, and so it kind of looks like theta, and then you get two circles when it meets on the other side. So it's going this way, and then here it's still going that way and this way, and then when it connects, it disassembles, and you've got two strands, or two circles. In eukaryotes, we have several replication origins, and so if this is a really long stretch of a chromosome, we might have some replication origins happening, all along, and then they'll just kind of grow into each other. And when they all finally meet each other, then it'll just be two strands. Yeah, I could have just shown you this picture, huh? All right, so here's a theta. And yeah, here's our multiple origins. Cool picture. All right, I mentioned that it grows in the 5' to 3' direction. And so we can take a look at this little diagram here and try to make sense of that. So this is 1/2 of the DNA, and these are two brand new nucleotides that are coming into this growing strand of DNA. And so what happens is something is moving along. We're going to call it DNA polymerase. This thing is moving along the 3' to 5' direction of the template strand, placing in 5' to 3' direction new chain, a new strand, daughter DNA. There's always going to be a nucleotide that's here to start, and it's going to have a 3' free hydroxyl group. So then the 5' phosphate of the coming in base, the new base that's going to be added to this growing chain, will always form our phosphodiester bond and then leave another 3' hydroxyl exposed. And so when they say that it grows in a 5' to 3' direction, that's all that means, is that there's always this 3' hydroxyl ready to react with the next 5' nucleotide. So since DNA can only grow from the 5' to 3' side, it makes sense what we just looked at for the template strand, right? We can go back real quick. It makes sense for this. Let's clean that up. It makes sense for this because as long as you're following this strand, you're always going to be making something complementary to it, and this is the exact direction that you can build a new chain in. So there's nothing wrong here. The problem comes with the other strand, right? This was separated. There was a fork that happened somewhere. I think the fork is probably more on this side, but this one is running, 5' to 3', right? Because it was the opposite of the template strand. So the new strand that's growing here would be growing in a 3' to 5' direction, and that's wrong. That we can't build it that way. So something creative has to be done to make sure that both of these strands, this strand and the template strand, are both being copied at the same time. And so there's a neat little trick that'll happen here. So we'll actually call the strand that we just looked at and we kind of studied the leading strand. That's the one where we can just build a new daughter DNA in the right direction, 5' to 3', no problem. The other one, the one that I just took you on a little adventure, we looked at and I circled a big spaghetti drawing. That's the lagging strand. Okay, so the lagging strand needs to be synthesized in these small little sections, and you'll see why in a second. The sections are called Okazaki fragments, or can be called that. The guy that discovered them or studied them. The 5' end of each fragment is closer to the replication fork than the 3' end. Fragments are linked together by the enzyme DNA ligase. And in most cases, any nicks in the DNA are actually going to be kind of taped together, pasted together with this DNA ligase. So that's one that we'll come back and talk about in a little bit too, when we talk about the repair mechanisms. So here's a nice sort of picture of it. So in order to get all of these Okazaki fragments that are, again, synthesized 5' to 3'. So this is the 5' end, 5', 5', 5'. They have to, this strand needs to be folded over. And so you can see that here. So every little bit that this DNA polymerase has moved down the DNA, something has to be folding over this lagging strand so that as this section right here is being written, this could be written at the same time. And then, we need to keep moving on, so then we need to let this go and now fold another piece and line it up. And so you end up with these just little segments, and they're discontinuous. Those get pasted together with ligase. Nucleoside derivatives. Your book talks a little bit about molecules or compounds that are not nucleotides, but actually have similar structures. These are used as, or these can be used as ways to kind of stop replication. So if you incorporate one of these, like an AZT molecule, into the DNA, normal polymerase will come around and it won't be able to read that and pair it with anything, and so then the whole process sort of just shuts down. And these are two of those structures. You can see some similarities. So this is specifically how it works. When those are incorporated, they don't form the phosphodiester bonds, and so the growing chain can't continue. So it just stops that sort of chain elongation reaction. Okay, DNA polymerases. These are the proteins that make the new strands of DNA. They are extensively studied, or at least 5 of them have been in E. coli, polymerase 1 through 5. Polymerase 1 is a single peptide chain, so like a tertiary structure. Polymerase 2 and 3 have multiple subunits, they're like quaternary structures. Recently, polymerase 4 and polymerase 5 have been found. Them, along with polymerase 2, are really more of like repair mechanisms, repair enzymes, as opposed to polymerase 1 and 3 that do the main copying of DNA and synthesizing of the growing daughter strands. They get kind of compared to each other in terms of their turnover or processivity, the number of nucleotides that they can incorporate. So let me show you that. You can see some differences here too in just their size. Polymerase 1 and 2 kind of on the same scale. Polymerase 3 much larger. This is our main one, by the way, polymerase 3. Prokaryotes main one. We have one that's analogous to that we'll talk about later. It's the most active, polymerase 3. You can see it's got quite a bit that it does before it actually comes off of the DNA. We know now there's actually 3 of these working on each of the replication forks. Let's see. This is neat. They talks about some of the activity. Now these have in prokaryotes some activity that we don't see in our eukaryotic polymerases. We actually have separate enzymes that will do some of these jobs. But in addition to these all being able to polymerize, they also have exonuclease activity, and that means that they can go and actually take out rather than just put in. Some of these can take out in the direction that they could write in, and some of them can actually take out in the backwards direction. So that becomes really, really cool. If the polymerase itself has incorporated the wrong base pair, it can actually go back and kind of cut it out and then retry. Let's see. There are some things that are required for DNA polymerase to actually do its job. These things were discovered just by kind of systematically leaving things out, leaving out components of the reaction and seeing, if it could still progress. So it turns out that if you don't have nucleotides present, that should make sense, that this reaction doesn't take place. It turns out, too, that polymerase can't just start making a copy of the DNA without something to attach that first nucleotide to. And so it needs something called a primer. And these primers, this is that RNA that we said at the very beginning would be associated with this process. So there needs to be these stretches of RNA that actually give a starting place for polymerase to start attaching the new nucleotides. And so for those primers, you got to have the RNAs available. Magnesium is required, and DNA template. That should be an obvious one. If you don't have a DNA template, then there's nothing for polymerase to copy. So here are some of those functions again. Polymerase 1, repairing and patching DNA. Polymerase 3, this is the main polymerizer. 2, 4, and 5 are repair enzymes. They have exonuclease activity that is for proofreading and repair. So proofreading is just putting in things that are taking out incorrect bases and sticking in new ones. And repair doesn't always happen from just the copying of DNA. Sometimes, like UV light, we'll see, can cause damage to DNA. And so then something has to go in there and repair that damage. And so these polymerases also have that job. So DNA replication carried out by something called a replisome. The replisome is just the complex of the polymerase, the RNA primer, the primase, helicase. All of these things, helicase and primase, are involved in just kind of unwinding the DNA so that it's available for copying. DNA gyrase, we talked a little bit about topoisomerases in the last chapter. This one also kind of goes ahead of the replication fork and kind of kind of super coils it, or super, what's the word that your book used? It accounts for the fact that as you try to split apart the DNA, it's going to lead to some supercoiling. So it kind of unwinds it a little bit up ahead so that when the supercoiling happens, it won't be so bad. So it's again there to help relieve some of those strains with the unwinding of this twisted DNA. We can kind of see again what DNA gyrase does here. Remember from the last chapter we talked about how it's used for introducing supercoils. Okay. Oh, here we go. This is what I was trying to say. So prokaryotic DNA is negatively supercoiled. Opening the helix during replication introduces positive supercoils. So gyrase fights against those by placing negative supercoils ahead of the replication fork. So in much better terms than I was able to deliver, that's what it does. Helicase is a destabilizing protein that helps to unwind. So you can see it here. Keeping apart, these two strands from winding back up again. Gyrase here doing its job to introduce some negative supercoils. We're going to talk about these right now, these SSBs, single-strand binding proteins. These protect, if you notice, the strands are split right here, but no new nucleotides are being added until way down here. So for this whole stretch of the lagging strand and this little stretch of the leading strand, there are there's single-stranded DNA, and things want to go and react with those bases. And so these are proteins intended to just kind of protect those. You can see here the Okazaki fragment being pieced together by ligase. You can see here in green the DNA primer, RNA primers, that need to be there in order for, I don't see the primer. Oh, we wouldn't see the primer. The primer would have been way over here on the leading strand. On the lagging strand, every Okazaki fragment is going to need a new primer. So RNA serves as the primer. Primase is the enzyme that makes those little short sections of RNA. And the primosome is the complex that consists of the RNA primer, primase, and helicase. So we had a replosome, we had a primosome, two different things. DNA polymerase 3 starts the synthesis. As the replication fork moves away, the RNA primer gets removed by DNA polymerase 1, and then DNA gets filled in rather than those RNA base pairs. So that's what this DNA polymerase 1 is doing. This is that exonuclease activity that actually works in the same direction that you're going. It can go in there and just read over those RNAs for that little stretch of RNA primer, cut them out, and lay in the proper DNA nucleotides. Ligase is still responsible for piecing all of those little sections together. And this is sort of a summary. It tells you everything. Could have just read this. I'm just kidding. You can read this, though. Your book talks about how polymerase is sort of like a hand, like your right hand, and that you can look at the different domains of it, like your thumb. If you were holding your right hand out in front of you with your palm up, your palm is sort of that pink area, your thumb is that green area, and then your fingers are somewhere in there. I'm not really sure why the fingers are these things and what all of this is supposed to be, but that's what your book says. Your book says it's kind of like a hand, at least in that they can, they can reference different domains, and they kind of know what each of these domains does. So there's a polymerase domain. This is the site of the DNA polymerase that actually makes the growing daughter strand. And then there's a different domain over here that's responsible for cutting out and kind of the repair mechanisms. It's kind of cool. Oh yeah, I mentioned that there are actually three polymerase 3s working on each of the replisomes. So one polymerase 3 is on the leading strand, and then there are two polymerase 3s that are actually on the lagging strands. So yeah, it used to be thought that there was just one polymerase 3 kind of doing everything, and now we see that there's a couple of them actually associated together. The clamp loader, this is really neat. This is the part of the molecule that actually opens up, kind of surrounds DNA, and then like latches on, like a little machine. And so they're kind of showing them here, clamped around the DNA strands. It's a pentameric enzyme, so there's 5 pieces, 5 different tertiary structures. And there is a different diagram somewhere. Oh, there's a diagram later that's going to show the eukaryotic version of this, because we have a slightly different setup. As far as proofreading and repair go, mutations occur every 10 to the 9 to 10 to the 10 base pairs. So that's not very often, but that's still, you know, your genome's long enough that that's going to be quite a few mutations if they go unchecked. And so, you know, since some of these can be lethal, it would make sense that nature would have devised some sort of proofreading mechanism. So as far as the proofreading goes, there's right after it gets inserted incorrectly, there's a little window where DNA polymerase can still take it out, put the right one in. Let's see, removal of the incorrect nucleotide immediately after they are added. So that's what proofreading is, sorry. And then errors in hydrogen bonding lead to errors in growing DNA chain. And so if the wrong base is there, then they'll have the wrong sort of hydrogen bonding setup. And that'll lead to there being, the pairs won't match up anymore. So those are the kinds of errors that can happen. So after exonuclease activity, which is basically, you know, being able to pull this out and put in the right thing before you keep going, we can get to the types of modifications that happen once the strand's already been made. And so there are a couple of different ones. There's the cut and patch process. This means you just cut out the whole mistake and you put in, you make a correction. And then there's something called Nick translation, which happens when you have a site of an error, and then a proofreading mechanism goes in there and corrects that error. There's a little cut in the DNA that gets moved to a new spot. So let's just take look at 2 examples of that. So this is just showing that sort of that initial activity. So let me explain. So here is a T paired with a G. Now that shouldn't be. It should be or a C. That's a C there. It should be a G. And so DNA polymerase, after the strand has already been made, comes in here doing its proofreading capability, and it'll find this mispair, and so it'll cut it out. So now there's a little nick there. And so then using its polymerase activity, it'll start right there, and it'll start to fill in. So I guess now we're down in this picture right here. So there's a nick where it took out that one little nucleotide. And so then follow the red strand, it's going to just build a new strand. It'll just rewrite this whole section. And then once it's, you know, rewrote that section, the error is fixed. It's had the effect of essentially leaving that nick somewhere else. So there is still a missing spot. Then DNA ligase just comes in and pieces those 2 together. Because remember, DNA polymerase can't join 2 branches together. It can only grow a branch off of, you know, using a template. So the mutations that we're talking about could be substituting incorrect bases, right? We saw that I think in a different chapter when we were looking at types of mutations. So there could be point mutations, frame shift mutations, insertions, deletions, that kind of thing. But there can also be damage that's done from outside agents, chemicals, sunlight, that kind of stuff. And a really common one is one that's caused by ultraviolet light. It causes pyrimidine dimers, so if two thymines are next to each other, these are often, they'll form a double bond with each other. And what happens is DNA polymerase won't be able to read, to pass that point, and so it'll have to just cut out that whole section and replace it. Now, I think this is another kind of example of that. Yeah, so here we've got two thymines. So this is one thymine, here's the other thymine, here's our DNA, and you can see there's now covalent bonds that have formed between the two bases. That shouldn't be happening. So that would be a mutation caused by an outside source and not just an error in copying. Oxidation damage, this is also something that can happen. And essentially, once the sugar gets broken, once this is the backbone of the DNA, so the strand will break when this happens. So this is another bad type of mutation. So few different repair mechanisms: mismatch repair, base-excision repair, nucleotide-excision repair, and non-homologous DNA end joining. So we'll look at that, and this kind of leads to some recombination. So mismatch repair is when you have two bases that shouldn't be together. So an enzyme that proofreads basically detects this, the mismatch is removed, and DNA polymerase goes back through there and rewrites that little section. Now the problem that arises is that how does your body know which strand is the correct strand and which strand is the incorrect strand, right? Because it's got a mismatch. And so this happens because immediately after replication, initially you have two strands of DNA, and your DNA strands are, I think they're either methylated or they're phosphorylated. They're methylated. So they've got CH3s on them. And so then after replication, right, you've got only one of your new strands that's methylated. And so if there's a mismatch detected, like maybe these two bases don't match up, the proofreading mechanism comes in here and is able to see which strand is the methylated strand, essentially, to detect which is the one that's the correct one and which is the strand that needs to be repaired. And there's a neat mechanism. You can look over the details in your book, but this is essentially showing you that. Base-Excision Repair. This is where the base, just the purine or the pyrimidine, is actually removed. And so, well, initially. So damaged base, for whatever reason, it gets taken out, and so now there's an apurinic or apyrimidinic site, AP, so it just means there isn't one there, a purine or a pyrimidine. Then something comes along and actually will cut out the rest of the backbone there, and then DNA polymerase comes in and fills in the gap, followed by DNA ligase to piece together the final little pieces of that. So base-excision repair starts by removing the base, and then ultimately leads to some of the backbone. Nucleotide-excision repair is what happens when just a big stretch of DNA gets taken out. Like we were talking about with the UV, there can be some chemical, like when we looked at the example with the AIDS, not AIDS, the HIV, HIV virus. How we can use those synthetic nucleotides to sort of go in there and just sort of bind up, and they're unable to react. Like if that happens, there needs to be a way to cut those out of the DNA. And so there's something called an ABC exonuclease. This comes in here and actually just cuts out the whole stretch, the whole big section, and then removes it. DNA polymerase 1 and ligase come in and repair that. Double-stranded breaks sometimes happen where both of the DNA just get cut. And so then this is where we have to, the DNA just needs to be sort of joined back together. And there's a few different ways of joining just two pieces of DNA together. One is non-homologous DNA end joining, and the other is recombination. So what we're looking at here specifically is a protein called KU7080. I think that's the name of it. It's a weird name. Yeah, it's a heterodimer protein called KU70/80. This thing just recruits some proteins together, DNA ligase, and it just joins two ends together. This thing is very error prone. We don't know what cut this. We don't know if there's missing nucleotides. We don't know if these were supposed to be connected or if now there's a big chunk missing. So this thing just hooks them back together. That's the point. Recombination is also another process. This happens. Whenever viruses sort of incorporate their DNA into ours, or this happens during some parts of mitosis when you're making actually like your gametes, right? Your eggs and your sperm cells. These get a little bit of recombination before they form to introduce some genetic diversity. So it happens in two flavors. We can get homologous, which is similar parts of the DNA, so sequences that are both, like, let's say, coding for the same protein, or non-homologous. This one is when completely different things kind of merge together. These occur within specific zones called hot zones on the chromosome. Here's one example of that. This is actually just physical exchange of chromosomes. So these guys are viruses. All of their genetic information gets into this same cell, and then it just sort of gets repackaged up kind of randomly. So some viruses just get, a pink portion, some get the blue portion, but some of them are going to get these sort of half and halves. And so that's the recombination in this sense. They're just exchanging parts of their actual physical chromosomes. Now, recombination, the way that it happens in our bodies, is a little more complicated. It involves some splicing of the DNA, some cutting, some crossing over, and there's a nice diagram here that's going to show it, but this sort of goes into a little bit more detail. And so here we can see the model that your book shows us. So here we have two sections of DNA. There's this one, and they are going to cross over. We get some nicking that happens, so places where we can now join them to each other. So like this light blue strand is going to come down and get connected to this pink. This one's going to get connected here, right? So now they are strands of their own. And then this branch migration is this little fork thing moves to a different spot, so it's a little easier for them to split. And then there's going to be a second cleavage event here. You can see this thing orients itself in a way that now this can all just get kind of cut right down the middle, and we're going to end up with this dark blue and this pink pairing up. I think we see that in this one right here. Anyways, you can see this is how DNA can kind of get mixed and matched. There's a few mechanisms for this. This is one of the ways that it happens. This happens a lot too, not just in the DNA that makes, or that during the production of your gametes. This also happens in our immune system when we're making antibodies. We want that kind of randomness, that variability that comes with being able to detect any foreign invader, even one you've never seen before. And so your body likes to really do a lot of mixing and matching. And so recombination like this happens in that process. Okay. Eukaryotic DNA replication is a little more complicated than for prokaryotes. For one, it takes place during a specific part of the cell cycle. The cell cycle is broken into these four different parts. There's the M phase, G1, S, and G2 phase. M is where the actual cell is dividing. G1 is when it's getting ready and building all the things that it needs to have DNA replication even be possible. Then there's the S phase where DNA doubles. And then G2 phase is that preparation for dividing. So we're going to go through what happens basically during the S phase and some parts of the G phase. Probably G phase and then S phase. Okay, so during the G phase, for eukaryotes, there are a lot more little interactions that have to happen so that the replication fork can form. And so there are these three proteins. There's these three associations of proteins. There's the origin recognition complex. So this is proteins that are bound to the DNA and control replication. There's the replication activator protein. These again bind and help this process get going. And then there's the licensing factors, the replication licensing factors. And these are essential for DNA replication. If they're not present, then DNA replication doesn't happen. And we'll connect that to cell division in a second when we get to a nice picture that kind of shows everything. Okay, so the pre-replication complex, pre-RC, is what we have when the DNA, the origin origin recognizing complex. Man, I always say it wrong. Yeah, origin recognition complex. When the origin recognition complex, the replication activator proteins and the recognition licensing factor are all present. That's what the pre-replication complex is. Now, this system is also regulated by some proteins called cyclins. Cyclins are proteins that get made during one part of the cell cycle, and then at a different part of the cell cycle they get broken down so that they're not always present. That way, when they're missing, they're not doing their job, and then when they are there, they are doing their job. So it's a way of regulating this process. Now, those cyclins combine with some kinases, cyclin-dependent protein kinases, to turn off and turn on, essentially, to turn on the replication event, but then to turn off the formation of new replication complexes. And so that's the kind of two jobs that it's going to do here. And there is a picture in a moment that I'm going to show you. So let's kind of show you, and then we'll back up and read that other slide. So here's early G1 happening right in here. So the origin recognition complex. Oh man, I'm going to say it wrong again. Origin recognition complex, perfect. So it's bound to the DNA, and then we get the replication activator protein comes and binds to the DNA, and then we get our recognition licensing factors, and they bind to the DNA. And so it's this process right here, when these guys get released. This doesn't happen because these guys are in the cytosol, okay? The DNA is in the nucleus, or the nucleosome. So when the cell is getting ready to start replicating DNA to go into S phase, right, to turn in from G1 to S phase, those membranes kind of dissolve. They go away. The nuclear envelope opens up. The DNA comes out, mixes in with the cytosol. That's the signal, basically the presence of those RLFs now being able to come in and start binding in these different spots for this process to start turning on. Now then at some point, cyclins would have been made, and so then now once all of these guys are here, the pre-replication complex, all four of these players, the DNA, and then RAP, RLF, and ORC, then the cyclin-dependent kinases start doing their job. When they come in, they phosphorylate these guys. That activates the process. It also dissociates these proteins. So these proteins now fall off. And they get degraded. And so this degradation means that they're not going to go and find some other part on the DNA and start doing, another replication complex. So that's the dual regulation here. It turns it on, but it also keeps it from turning on in other places. And then we have our replication then happening during S phase. And so you can see it's bidirectional. It's going to go both ways. And then by G2, we've already got our copied DNA. A lot more polymerases are present and been discovered in eukaryotes. A lot more than have been studied, for prokaryotes, though we do have some that are analogous to those. primarily it's going to be our, let's see, our delta. I think our delta is the one that's most like polymerase 3. We'll see here in a second. We do have some differences. We have one type of polymerase that's exclusive to mitochondria. We don't see it anywhere else. We do see that a lot of our polymerases do not have exonuclease activity, unlike what we saw with the prokaryotes. Yeah, we also don't seem to know if this has repair capabilities, at least not for delta. Polymerase alpha makes primers. Polymerase delta is our main one. Is that the one I thought? I think that's the one I thought. Polymerase epsilon, it says involved in leading this in the leading strand replication, and it may replace polymerase delta in lagging strand synthesis. Looks like beta is our repair enzyme, and gamma carries out DNA replication in the mitochondria. So some of the exonuclease and proofreading activities then have to be kind of offloaded to other proteins and other enzymes in eukaryotes, because our polymerases are, it's a more complicated picture. They don't seem to be doing all the same jobs. So in eukaryotic DNA, or in eukaryotes, polymerase 3 doesn't exist, right? Polymerase 3 is in prokaryotes. Polymerase 3 had on it in its structure the part of it that clamped on to the DNA. In our bodies, we have polymerase alpha, which is essentially, polymerase delta, which is the main polymerase. It interacts with a protein called pCNA, and it's like the equivalent of the part of POL3 that functioned as a sliding clamp. It's a trimer of 3 identical proteins that surrounds the DNA, and it basically works as that clamp. There are some differences, some notable differences in DNA replication from the 2 different types, prokaryotes versus eukaryotes. Some similarities and differences. As far as some of the functions of the polymerases go, they don't all seem to do the same types of jobs. In prokaryotes, we see that the polymerases can do exonuclease activity, but we don't see that necessarily in all of the polymerases of eukaryotes. We do see differences in terms of the replication sites. There is only one origin for prokaryotes and several for eukaryotes. Of course, there are histones associated in DNA of eukaryotes. And then the Okazaki fragments are also much longer in the prokaryotes than in the eukaryotes. So those are a few of the differences that we see. And here are some of the basic layouts of the eukaryotic replication fork. You see not too many differences here. So a topoisomerase goes and supercoils it, gives it a negative supercoil so that when it unwinds, it's not so strained. Helicase here helps to keep it unwound. RNA primers on the lagging strand for our DNA polymerase alpha to work on. We've got some ligase also working in here. We didn't talk about all of these other little like FEN1 and RNase H1. Basically, RPA is just the eukaryotic version of the single strand binding proteins. So these are just the and this FEN1 and RNA ACE, is it FEN1? Yeah, FEN1 and RNA ACE, these two here, these guys have to go in here and actually get rid of that RNA primer. They degrade the RNA because the our DNA polymerases don't have that exonuclease activity, so they can't cut anything out like in prokaryotes. So that's what FEN1 and RNase H1 are doing. And then did we leave anything else out? Let's see. There's pCNA. What does pCNA do? Oh yeah, PCNA was the clamp that slides on it. That was the molecule we just looked at right up here. All right, the last bit has to do with your telomeres, and your telomeres are these long stretches of DNA at the end of your actual coding DNA sequences, and these are usually a series of repeated DNA. Non-coding, it acts as a buffer against degradation, so that if there are chemical reactions eating away at the ends of the DNA, your genetic code is safe. Another thing that they do is they actually provide a surface, right? If your DNA just ended and you were trying to copy that, DNA polymerase, right, and all the little bits that it associates with don't have anything to hold on to at the end of the strand to actually copy those last couple of base pairs. And so it makes sense to have a long stretch of just, DNA that this thing can actually move on to get the last bit of that important information copied. There is some information or some research out there that has related telomeres to cancer cells and related telomeres to basically being younger and living longer. So there is a lot of information. I keep saying information. There's a lot of research going into studying telomeres, and your book does have a neat little relationship, a biochemical connection between telomerase and cancer. You should take a look at. But I won't be including that on the test or anything. But if it's interesting and you're interested in that, it's definitely worth the read. It's very neat. All right, I guess that sort of wraps up Chapter 10. I'll be posting a discussion board for you guys to put any questions you have in here. Really just focus on sort of the big picture ideas, some of the similarities and differences between prokaryotes and eukaryotes that we talked about, and just the different mechanisms for repair and the different complications that arise when we're copying DNA. Like how do the strands unwind, and what do we do about all that that supercoiling, and how do we protect the single strands, and how do we take care of the lagging strand? Like those are the kinds of ideas and topics that you should focus on